So good day to each and every one of us. I hope you're doing fine. I hope you're coping well with the stress of this online learning as well as the uncontrollable increase of cases of COVID. Um, Ayun, stay safe pa rin, no? And ayun, let's go to our topic on continuation of our topic on DNA. So basically this week we're going to talk about eukaryotic DNA replication. It follows that when we discuss prokaryotic DNA replication, we also need to talk about eukaryotic DNA replication because this is where we belong, because our cells are eukaryotic. And then there are just some notable differences which we will take note later on as we progress. And then ayun, we will also going to talk about DNA repair for this session. So for eukaryotic DNA replication, the process of eukaryotic DNA replication closely follows that of a prokaryotic DNA synthesis, with just some notable differences like your multiple origins of replication, because our DNA is linear as compared to your prokaryotes, which has a circular DNA. Another difference would be your polymerase. So it is if our prokaryotes are termed DNA polymerases 1 and 3 or 2, our eukaryotic DNA polymerases will have a Greek letter assigned to them. And then another one would be the RNA primers. So for our eukaryotes, the RNA primers are removed by your RNase H and FEN1. As compared to your prokaryotic DNA replication, our RNA primers are removed by what enzyme? Your DNA polymerase, specifically DNA polymerase 1, diba? So these are going to be our topic for today, the eukaryotic cell cycle, the eukaryotic DNA polymerases, telomerases, the concept of reverse transcriptases, and the inhibition of DNA synthesis by nucleotide analogs. So let's talk about the eukaryotic cell cycle a bit. Okay, I know that you have tackled this in your undergraduate, so medyo gas-gas na to, no? But it is important to note that in eukaryotic cell cycle, it goes hand in hand with our DNA synthesis. Okay, so as this cell cycle is proceeding, our DNA synthesis actually occurs as the cell cycle is occurring. So they are in a coordinated manner. So as you can see here on the picture, no, so we have here your gap1, and then enter siya ng DNA synthesis na part, and then your gap2, and then for your mitosis. There are some cells that would actually not divide, diba? So they would go to G0. This G0 phase are those cells that have cis division. So these are your neurons as well as your muscle cells. And then if sometime your actively dividing cells would reactivate, no, it will enter your G1 again, and then DNA synthesis, gap2, and then your mitosis. What's important to take note here is that the cell cycle is controlled at a series of checkpoints that prevent entry into the next phase of the cycle until the preceding phase has been completed. So this is in order for us to control, no, the uncontrollable proliferation of cells, which would eventually lead to cancer. And then the two key classes of proteins that control the progress of a cell through the cell cycle are the cyclins and cyclin-dependent kinases. So these two proteins, they are your main regulatory proteins. So for your eukaryotic DNA polymerase, okay, wait lang. Let's go back here muna sa eukaryotic cell cycle. As a review, no, the period preceding replication is called your G1, and then DNA replication occurs during your S phase or your synthesis phase. Following that, there is another period which is your G2 or your gap2 before mitosis, which is your M. Okay, and then cells that have stopped dividing, such as your mature neurons nga, and then your muscle cells, they are said to have gone out of the cell cycle into the G0 phase. And then cells can leave the G0 phase and then re-enter in the early G1 phase to resume cell division. And those cells are your actively dividing cells, like the cells on your skin, which are on your stratum, located on your stratum basale or granulosum. Those at the bottom part. For your eukaryotic DNA polymerases, there are 5 key eukaryotic DNA polymerases. Okay, lagyan nyo ng polymerase yan. And then they are designated by Greek letters rather than Roman. So they are categorized according to molecular weight, cellular location, sensitivity to inhibitors, templates or substrates on which they act. So we have here a summary of that, okay, of your DNA polymerases. So basically, your polymerase A, it contains a primase and it initiates your DNA synthesis. But then it doesn't have a proofreading ability. So wala siyang exonuclease, 3 to 5 exonuclease property as compared sa prokaryotics na meron, diba, yung DNA polymerase 1 and 3 natin. And then for polymerase B or beta, it is mainly for repair. And then for polymerase gamma, it replicates mitochondrial DNA and it has proofreading ability. And then for your polymerase delta, it is thought to elongate Okazaki fragments of the lagging strand, and your epsilon, it is thought to elongate the leading strand. One thing that serves me to remember this delta and epsilon is that your leading, it has an E, so which makes it epsilon. Diba? I want you also to take note that in polymerase A, it is a multi-subunit complex. Okay, when we say multi-subunit complex, your polymerase contains the primase as well as the necessary enzymatic reaction that would elongate the DNA strand. Okay, because as compared to your prokaryotic DNA synthesis, the DNA primase or your RNA primase or your primer is initiated or made by your RNA primase, diba? So another enzyme is responsible for the incorporation of a primer. As compared to our eukaryotic DNA polymerase, polymerase alpha already contains the primase which will initiate the making of your primer. So now let's talk about your telomeres, okay, the concept of telomeres. So your telomeres are basically yung nasa, if you recall the parts of a DNA, diba? They are basically those at the extreme points of your chromosome, diba? They are at the most extreme parts. So your telomeres are complexes of non-coding DNA plus proteins located at the ends of linear chromosomes. Basically, they function to maintain the structural integrity of the chromosome, thus preventing the attack by nucleases. They serve as a cap. Ganun ko siya inaalala. They serve as a cap so that yung mga nucleases, they will not damage the DNA. And then they allow repair systems to distinguish the true end from a break in a double-stranded DNA. So let's talk about the human telomeric DNA. So your human telomeric DNA, it consists of several tandem repeats of an encoding hexameric sequence, which is our AG3T2, which is your AGGGTT, just repeating na mga ganon. So AGGGTT, 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 that would be the composition of your telomere. It is base paired to a complementary region of cis and ace. Malamang, di ba? Kasi yan lang naman talaga yung complementary base pair ng AG3 and T2 natin. So for your GT-rich strand, it is longer than its CA complement, leaving a single-stranded DNA a few hundred nucleotides in length at the 3' end. Later on, you will see this in a picture. And then the single-stranded region is thought to fold back on itself, forming a loop structure that is stabilized by a protein. So let's watch a video regarding telomeres, okay, so that we can fully understand its structure as well as its function, because it is involved in cell senescence or cell death.
What are telomeres? Telomeres are structures made from DNA sequences and proteins found at the ends of chromosomes. They cap and protect the end of a chromosome like the end of a shoelace. In almost all animals, from the simplest to the most complex, telomeres are required for cell division. With each cell replication, the telomeres get shorter and shorter until they're so short that your cells can no longer divide. When cells no longer divide, tissues age. However, telomeres can be rebuilt by an enzyme called telomerase to restore cell division. If you think we've found the fountain of youth, think again. If a cell keeps dividing uncontrollably and overcomes the limitations of telomeres, a cancerous tumor can form. This is why telomeres are so important. In healthy cells, they achieve the necessary balance between limiting cellular lifespan and your own longevity and keeping cancer growth at bay.
So there, as you have heard, diba, your telomeres are basically one of the structures din that would actually prevent a cell or a DNA to uncontrollably synthesize or replicate itself. I think I still have another video here on a molecular basis or the basically the structure on a molecular basis.
Telomerase function. The ends of chromosomes contain regions referred to as telomeres.
So that is the overhang that I was talking about on the previous slide. Okay, that overhang serves as a template for your telomerase so that it can extend the DNA. Okay.
Telomeric regions consist of telomeric repeat sequences. The exact sequence of telomeric repeat can vary from species to species. The repeat sequence shown here is found on telomeres of human chromosomes. Since DNA replication can only proceed in the 5' to 3' direction and requires a primer, it is not possible for a DNA polymerase to replicate the 3' ends of linear chromosomes. If this problem were not corrected, chromosomes would shorten with each cycle of DNA replication. The addition of telomeric repeat sequences by an enzyme complex referred to as telomerase assures that this shortening does not occur. Telomerase is a unique and interesting enzyme because, in addition to having DNA polymerase activity, it also contains an RNA sequence that provides a template for the synthesis of telomeric repeat DNA. Part of the RNA sequence hybridizes with the single-stranded overhang on the DNA strand, leaving a single-stranded overhanging RNA sequence. The repeat unit shown here is from Tetrahymena, which is the model organism used in telomere and telomerase research. The DNA polymerase function of telomerase then synthesizes a DNA strand complementary to the RNA found in the telomerase and subsequently translocates to the end of the newly synthesized strand, and the process repeats itself. Many cycles of repeats can occur. Once telomerase has completed its function, DNA primase synthesizes an RNA primer near the 3' end, and DNA polymerase fills in the vacant region. A short region at the 3' end will remain single-stranded. The end result is that telomerase will have added many repeat sequences from a few dozen to a few hundred. This prevents chromosome shortening.
Ayun, nakita nyo naman na yung overhang na yon, it would actually serve again as a template for the telomerase to attach itself so that hindi mag-shorten yung DNA. Okay, I hope, ayan. This is a nice picture telling us the function of the telomerase. Okay, again, on this part, so the telomerase extends the 3' end of the DNA. Yan, ito yung overhang na nakikita nyo dyan. And then the RNA primer is synthesized by the primase, and then the 3' end of the RNA serves as an acceptor for DNA polymerase, and then of course it will synthesize a newly synthesized strand, and then the RNA primer is then removed, leaving us a overhang of an AGGGTT sequence. Okay, so now let's move on to the topic of